honor to have with us Arne Elofsson, who is a professor of bioinformatics at the Department of Biochemistry and Biophysics of the University of Stockholm. After his PhD in medical biophysics at the Karolinska Institute, he moved to Heisenberg Lab at the UCLA University in California, and from there he got back to Europe in 1995 to the Stockholm University, where he became full professor in bioinformatics in 2006. And since then, Arne had very many interests in, again, in structural bioinformatics, and particularly on proteins and their function and evolution. Through the years, he developed very popular tools that we all have been using for the prediction of structural and functional features of protein, including membrane proteins. More recently, became interested in embedding problems, as we heard yesterday, including deep learning applied to protein interaction. Just to mention a few of his most recent publication, to give you an idea, for example, title are as such: intrahelical salt bridge contribution to membrane protein insertion, modeling SARS-CoV-2 proteins in the CASP commons experiments, protein sequence to structure learning, is this the end-to-end revolution, etc. So thank you for being with us, Arne, and it is a pleasure for us.
Thanks for having me. I think the easiest way is I will give sort of maybe divide my talk into like 2 or 3 or 4 sessions, and then after each I will give you a bit of like time for questions, and I appreciate questions because it's but it's a bit difficult to interrupt in the middle because I will not see your hands going up and so on. So I think the easiest is if you save your questions until I ask you to do it, and then I'll stop a bit and then I'll try to look at you, and hopefully you have some good questions. And I will start with a bit of historical background of basically the field and our work, but also others' works. And then I will try to actually end with things that are not, that I got the manuscript first version of it last week. So it's going to be all the way from things happening almost 10 years ago until today. So I'll share my screen. Hopefully it works if I do it like that. And I view slideshow. I think you see my screen now. Do you? Yes. Well, otherwise you have to scream. So basically, we are all, of course, you all know about AlphaFold, everything's happening right now. But I will, anyways, take you maybe 10 years back to think it's how did it start? And we have to think about data. And just 2 old data point entries, and this is a log scale, and this is some years in the beginning. And you have 3 curves. You have the PDB curve, and the uniprot curve, and the PFAM curve. So uniprot is red. So uniprot has a clear exponential increase. I think it's continued even since 2017. It's basically very, very exponential. PDB sort of had an exponential increase, but it's flattening, and particularly the exponential growth rate is much lower. So the gap between these 2 curves are sort of is increasing. And also important, if you look at PFAM, so the number of families in the database, it was created and it was grow fast, and then it's basically been almost flat. And the number of new PFAM families entering every year is quite low, and the coverage is still about 75% of all proteins, something like that. So the main parts that are not covered is, I mean, some unique sequences that probably exist in a very, very few organisms, but a lot of disorder, very fast evolving regions and so on that are not covered, and maybe some repeat regions also and so on. So there is probably, excluding this type of regions, there probably is a limited amount of protein families existing in nature. So if that's 10,000 or 100,000, we could discuss, but it is not exponentially increasing, at least with the number of new sequences. So what does this mean? We have an exponential curve and basically a non-exponential curve. That means that the number of sequences for each family or each structure or structure family is increasing very rapidly. So this is old data. So you saw until about 2010, most families were small. You had 100 sequences or 200 sequences, so on. Like they were not maybe 500, but then after they just taken off. So now, of course, we have in most PFAM families we have tens of thousands of sequences there. First of all, this has been a challenge then for all bioinformatics tools, because how can you handle it? A tool that can make a multiple sequence assignments of 10 sequences cannot do it for 100,000. And so, or even searching these databases or clustering them is getting hard. So there are, people have now developed extremely good tools to do that, but that's not what I want to talk about today. But it's clear it puts the challenges, because this growth rate is much faster than computational growth rate. So you need to have better methods. And particularly as many, and we want to clustering in this case as a square also. And so it's basically impossible to clustering unless you have a very, very good algorithm. But the important thing here is that we have much more information about each family. So that means if we can use this information, we can learn something. And the information can be presented in some sort of multiple sequence alignment. So this is the key thing. And of course, normally, historically, we do it, look at one column in this alignment by one column. We look at if it's conserved, we look at what amino acids are there, and if they have insertion gaps. And that, of course, can tell you a lot about the structure of protein. We can learn predict secondary structures. We can learn predict loops, if it's on the surface, non-surface, memory regions, etc. So that cannot. But that is sort of an averaging approach. We are looking at not using the whole multiple sequence alignment, we're using some sort of average column by column. So the first step of going beyond that is to look at pairs of columns. So if you take one column here and one column here, and you look at, is there a relationship between these two? And the easiest way to look at it is to look at correlations or mutual information. So basically, the classical example is if you think you have two amino acids that interact, one is small and one big, and then this big one mutates to something small, and the small one can mutate to something big. So then you should have some sort of a correlated mutation. But it can be positive, negative charges. It can be more complicated. So that was an idea that I guess mainly Alfonso, that you heard yesterday, introduced in the mid-90s, so like most likely before most of you were born. Not me, but others. So that was a long time ago. But it didn't really work. It didn't really provide any information. At least it didn't provide any structured information. I mean, it provided some information. It was better than random, but nothing really useful. And one reason for this is that it boils down to a problem in statistical physics of spin glasses in the mid-90s, and where you have indirect and indirect interactions. So if you think about a problem like this, so this is an idea how a protein looks like. It's like some parts of the central protein that is interacting, and you see things are very well packed. You have like nice interactions, they're packed nice to each other. You have it's well evolution on it, and then you have a mutation. That's this pentamer mutation arrow, and of course the packing is not as good. So that's so you will lose some energy probably, but maybe has some other benefits. It survives evolution anyhow, or it's just neutral. It doesn't really matter, but at least the mutation happens. But then it's the pressure to do a compensatory mutation. So then you have another, I guess it's a cube mutating to the pentamer. So then you get nice packing again. So this is the co-evolution feature. But if you look at the top of this arrow, it's not the same as the top of the pentamer. So you have actually parts over here that are not, that have changed also. And of course, all amino acids in the protein are coupled to each other. So if you think about it, it's a big network of interactions. So one change in one reduces many, many other changes. So this is what we call the indirect couplings. So you have a lot of couplings and indirect. And this mutual information, if you look at pairs, doesn't take that into account. So then people introduced, Martin White primarily, but also other people introduced it's called direct information contact prediction methods, DCA methods. There were also some other Bayesian methods. Originally, this is Lapidus from 1999, who never didn't work because we didn't have any statistical data in that case. But then in 2008, 2009, there was a number of papers that come out doing this. And they basically developed methods that you separated the direct coupling analysis from the indirect ones. So you have a target function you try to optimize, and you have this type of, and you can solve it, and you can find more of the direct information. So this is an example. So this is a contact map. I'm sure you've seen it before, but I will explain it anyhow. So you have the sequence of a protein here from 0 to 270, and then on the other axis, the same sequences. And every gray dot here is a contact in this protein, and you see that they have certain patterns, and this is the protein over here. And then the blue predictions in this case are correct. The green ones are almost correct, and the red ones are wrong. So this is the prediction. So the blue and green and red is the predictions you get out from a prediction method. So in this case, if you use this mutual information, you have 22% of the prediction correct, which is pretty good for mutual information. It's better than you normally get. And in this case, we do one prediction per residue. And that is actually less than the total number of predictions should be. So you probably should do more. But it's actually an unusually good prediction here. And you can see there are some contents here that actually have a lot of information. And there are some contents that are correct. Most of them are close to the diagonal, which is not so informative, but there are some other ones that are good. But there are also a lot of false ones. So there's some information there, much better than random, but not really useful. If you instead take this the same type of methods, so here I compare two of these, so PsyCov and PLM DCA. So they are methods PsyCov developed by David Jones and PLM DCA by Ekeberg et al. You see that you jump from 22% to 52, 58%. And you can see that you have really blue patterns coming down here, predicting things that are far away. I mean, they are not perfect. It's only half prediction correct, but it's not too bad. And you could imagine that if you had this information, you actually could predict the structure of proteins. So that was shown by Debbie Marks and also by Martin White and Terence Waugh and other people, that you could do that for many small proteins. You could basically take this information, put it into some kind of folding program. You can use CNS or Rosetta or whatever you want, and make quite good predictions. So now enters the, so this has happened, and there was some development, people did this in the statistical methods. Now enters the era of machine learning. So from a machine learning perspective, what do you think about when you see this map? Well, it's an image, basically. Machine learning has had also a gigantic leap in performance starting like 2008 also. By image recognition, so you recognize cats from videos or whatever they are. And what they do is, of course, they look at non-randomness in maps. And this map is clearly non-random. It's not like random dots separated, because the protein structure puts constraints into the how the content map can look like. So these constraints, of course, makes it non-random. And of course, computers are extremely good at finding patterns. So you can train something to find these patterns or describe these patterns. So we did this. So now we go back historically. We were one of the first people who did this in I don't know, 2012 or something. And the idea here, if you look, you can just think at a very, very simple pattern. What you do here, if you take one point in this content map, you take a random point over here, wherever you are. And then you have 8 surrounding points. So you have like residue I and j, and they have I in contact with j plus 1, or I minus 1, and I j 1. So you have this. And you can divide these points into 2 groups. The ones that have a contact and the ones that do not have a contact. And then you have in total 12, you have different patterns of other things. If you have a contact in the middle, you have no contact surrounding it. You can have one in this position, or one in this position, or one in this position, or one in this position, etc. But and if you have no contact in the middle, you can have no other contacts at all, or you can have one in the different positions around it, or two over here. So you can see that for first thing, you can see in both cases, if you have contact, no contact in the middle, the most frequent pattern is the one which is up to the left here, is the one that has no other contacts. But then you have like one contact in here, there are like one contact in the word, basically you have all options here. But here you have 4 patterns of one contact, and then you have actually 3 contacts. All the diagonal 3 contacts, and they have diagonal 2 contacts later here. Well, so basically, you see that patterns are different. And more interesting one is to look at really the frequency of this pattern. So this is if you just sum up all the neighboring, how many contacts you have in total in these patterns here, and the frequency of these. So here you have 4 contacts, here you have 1, here you have 2. You can see that if you have a contact in the middle, basically having 1 contact, so basically no other, or 1 extra, or 2 extra, 3 extra, up to basically 8 extra. Some of these are basically flat. They're basically 10% each, more or less. While if you have no contact in the middle, basically everything is empty. So you have like 99% is empty. You have 1 contact is like, 1% or 1.5% or 2% maybe. And then 3 contacts is less than 1%. And that's all the 2 contacts together. So basically, contacts likes to be close to contacts. As we see here, you have contact here, you're going to have a contact here, and preferably a bit diagonal also. So we used a very simple machine learning method to do this and developed what we call PCOC2. So in this, but it's conceptually a bit similar to what people did in ESA, but we used random forest. It was not advanced deep learning, but it had the idea of deep learning because we had an iterative step here. So in this case, we did predictions using the SciCov and PLM-DCA, have different alignment methods. So we had in total 16 different types of contact predictions. We added information from some secondary structure predictions, and then we did this method. So and it's basically a machine learning method that also can take into account the predicted contacts, so you can sort of like iterate through it. So if you look at layer 1, and this is just one example. So this first layer, look up in the left. Our predictions are 36% accurate. It's not that bad, but it's basically what you have. And you see you have this diagonal here, but we had a lot of false positives up here in different cases, red ones in this case. And then we go to layer 1. And you see, we are doing better. So like after 1 iteration already, we have 50% correct. You have more green contact in the right places and less false positives. And then go on, and you have layer, sorry, I did the wrong plot. So this, if you go up to layer 4 or 5, you end up with like actually up to 70% accurate contacts. So basically, you fill out this form here, you get rid of all these false positive ones, and you add more contacts here. So like you don't have everything, you still things missing, but it's much, much better than it was before. So basically, it's iteration. But and we used this in CASP 12. So there was one target. We were not winning CASP 12, but we had one target where we made the best model. So you have here was like target 0872, and we made a very nice, in our mind, a very nice product. You see, it's quite accurate, the secondary structure. This is a de novo target, nothing really homologous, anything else. And the loop here is certainly not perfect, and the secondary structure is not perfect, but it's at least for 4 years or 5 years ago, it was pretty good. And also blind prediction. So it worked in some cases, and the contact layer nicely, and it was also like a bit of help. I think Rosetta in this case liked this model because it packed it very nicely. Okay, then we and others basically jumped into the era of deep learning. So our attempt was PCOC4, so basically we use something called UMAP, where you have, but the idea is very similar to what everybody else is doing. So you take your sequence and some information from your sequence profiles, and then do a cross product, and you put that in, and then you add, so you can basically, everything in sequence prediction you have, you get also into a map. And then you add the information from Gauss DCA, mutual information, etc. And you add these two layers, and then you have using some techniques that are used from image recognition to basically predict new contact maps. Or in this case, it's new we even predict distances and different contacts here. And so in this case, we use UNet. When the other people use other methods that probably are better, but we could see some significant improvement. And particularly, our improvement was important for protein of small size. So this is a plot where we have the effective sequence, so basically size of the multiple sequence alignment. And you have the different methods here. I will explain them in a second. And then you have the accuracy in PPV, so the fractional correct predictions if you do one prediction per residue. And you can see that these methods here that are dashed, they're all these DCA type of methods. So they are pretty good. They're up to like 50% if you have 10 or a few thousand sequences. If you take this red line here, which is phi-C map, which is a method that does not use this direct coupling, it's just traditional machine learning method, it's kind of flat. It doesn't matter if you have 30 sequences or even, in this case, even 5 sequences or 5,000. It basically doesn't matter. And it never reaches over 30%. It's okay, but it never reaches more than 30%, which is borderline not enough to make a prediction. But it's not that. It's independent. But if you combine the method of this DCA, split machine learning, you get performance here on top. But you also, so our method is the black one, MetaPsycho with David Jones, another one, and our old method was this one. And you can see that you have, in particularly in the small multiple sequence alignments, when you only add a few hundred sequences, you get a big improvement. Up here, it's still better, but not by such a gigantic leap. So really, the machine learning could use things less efficient. More efficient. And well, this was the performance. We jumped up from like PLM level like 30% up to 50% with the earlier methods, and then 65% were there. And we developed some other things we thought were important in those times. We developed some database. We did this draft score, and we developed some scoring functions we thought were important. We tried to tune this. Can we do accurate predictions? So can we know that we were right? And it was not so good because it was like there are cases with many sequences we do wrong, and there are cases where we say number of sequences increased, but it's this large spread. Like sometimes with few sequences we do it well, sometimes with many sequences we do it bad. And if you combine these scores here, we could actually get a better ability to separate good from bad predictions. And we could do this, and we had, both we and David Baker had papers more at the same time, where we could predict a few hundred structures in PFAM that were not known at the time. So there was at least some biology out there. The models were maybe not perfect, but at least they could give you a hint about the structure. And we had to allow quite high false positive rates here. We had probably 10% false positive rates. Otherwise, we didn't find that much. So it was useful, but not really into the quality that biologists want. And this was just shown that Baker's and our models look the same. Their models are slightly better, but we basically in general most agree. But we were complementary in the families we found. And then I guess the key paper here was this by Jimbo Cho. So if you want to read the paper, you should read this one. And the idea is similar to what we did in PCON-C4. It's just the results are better. So they have sequence profile structures, so what we call 1D information. They do a cross product of that, add PLM, DCA, and other type of predictions that are contact predictions. They merge them together, and then they use 2D residual networks. The residual network is clearly better than what we did, and the predicted contact map. And that contact map was, so the key element is the ReLU element. I think that David talked about that a few days ago. So I will not go through that again. And this is their methods. Basically, their methods are right. It's basically every case they look at their better than alternative methods, the earlier methods. They had a significant improvement. And if you look at the models they made, it looked like that. So it's a bit similar to this one I showed before. It's okay. Loops and the regions are not very well perfect. And sometimes the helix is the wrong way. But in general, you have the secondary structures quite nice aligned. But it's sort of like the fold of structures. No details there you couldn't do. So to get biology from it is hard, I would say. But it's certainly a very good prediction. So this was a blind prediction in Camille in this case, and it was much better than anyone else this time. And I mean, some other cases, you see you have severe mistakes, like this helix here is wrong, completely wrong orientation, but some are better. So it's in a few cases it's very useful, but it's not in general. So then we entered the era of DeepMind, and that was in CASP 13. So in CASP 13, DeepMind did, in principle, there was nothing major new. They basically, but one major difference is the distances. We had done that before also, but not in a very good way, not as efficient as they did. The inputs are very similar to what we did before. They had much better networks, much deeper networks than anyone else had before. Partly because the limit to like small cells, and partly maybe because more training capacity, but it really made it much better network than people done before. Yeah, and then they had some, and actually a very simple minimization protocol they used. And that really jumped performance. You can look at this is CASP performance. So this is the often way the CASP presents the results. So they divide the targets into easy to difficult according to how similar it is to anything in PDB, basically. And that's some other criteria, but they have some criteria that is sort of, you can scale by different CASPs. And then you have the accuracy here. And you can see that you have, like, you start with CASP 1, and you had basically, still for the easiest target, you have very good models, and then it dropped quite quickly. And you can see that for CASP 1, 2, 3, and up to CASP 4, there was a clear improvement of the medium accuracy models. But the hard ones was basically, also CASP 1 is basically the same. There were a few targets where David Baker or David Jones or someone could make a good prediction, but what good was like, you got the fold right, but not more than that. And then there was really nothing major happening here. Until CASP 12, we started to see some improvements again, but here in the hard ones. Until CASP 13, we decided jumped up to having 60% GDTS. So there was a clear improvement. And basically one of the contribution was here that you had, this is the predicted distances. So you see, I guess the top line is the truths, and the bottom line is the predictions, and you see that quite accurate, maybe not in the right one, where you have some missing residues, but the right ones, the up ones are very, very similar. So you even have contexts that are far away at the end that looks nice, and you have things that look very nice. And you can make models that are almost starting to become biologically useful. They are, at least one to write here. It loops is not perfect. Here this is there, but in general, you get even folds of two sort of subdomains together, and you get things that work quite well. And soon after CASP 13, David Baker and Sergey Oshinikov and others released TIA Rosetta. That is very similar. It has a different, it also predicts angles between the interacting chains that maybe adds a little bit, but you can use that, and then they turn these histograms into residue restraints, and you use a simple method to minimize these models, and you do some relaxation, and you have a final model. And it was a nice thing that it was available. Anybody could run it. It was quite easy to run, not super fast, but at least it was not, I mean, it took a couple of hours or something to run a model. It was clearly very nice to use. And the models are, I mean, similar in performance as the deep AlphaFold 1 models. And so basically, if you run this on a set of models, you have like models that look like that. You see, it's not perfect. The side chains are not, loops are not perfect, but most in general, you have the secondary structural elements correct. And this is just some average benchmark, different things. And you have, most models have like a team score of like 0.7 or higher if you do this. I mean, there are always a few cases you fail and things like that. We had a special interest, we had problems before. We had repeat proteins. That was a problem. So if you use these DCA methods in repeat proteins, you have these kind of artifactual errors in your contact maps, because basically repeats are repeated, so you got things that. But the machine learning methods gets rid of these. They learn it. In particular, even PKC4 does it, but even deep intercycle and TRZ does it. This means that you can predict quite many of these repeat proteins quite accurately. Not all, not like the one, the DAF4116 seems to be wrong, but most of the others actually get nice predictions, even for spanning several repeats. So that was something we were happy about. Okay, now I thought it was a good time to have a little bit of talk. So like, I guess I would start asking, so if you go back to CAS 13 or like before CAS 14, so what do you think was the most important? I mean, what really did it say about what information was used and what was important contributions there? I mean, how much was it that data increased? How much was the machine learning, do you think? Or is it something that you think that is something else was important that was contributing to this performance? And so on, yeah. So do you have any questions about that or any comments? You can either just speak out or raise your hand or write in the chat. So it's question was the difference between helix and the beta strand. One surprise to me was that actually, I mean, you would, because I would have thought that beta strands are very hard to predict because basically all this coevolution happens between the side chains. So, and in beta strands, the interactions are mainly between the main chains. You have really main chain interactions that are binding, but so I thought that beta sheets would be very hard. But we had a paper where we did on beta barrels, out of beta barrels, and the predictions, even though the DCA methods were excellent. So it might be, so I don't, so in general for the predictions context, I don't think there's a big difference. There was some methods had some bias, but in general they were both quite good. And almost like the beta was slightly better. If you look at the folding methods that had people in the beginning, the betas are often harder because you have more long range information, so you really have to get things together. So, but in general, that was not a big difference, and that is different compared to earlier or this kind of fragment-based methods where alpha helix was always much easier. So that was a big difference. Anything else? I mean, I can just reflect myself. So I was impressed by these things, but it was sort of like when we were using it, I mean, we had some predictions we did for collaborators. I mean, someone in the lab had been working on protein, and we made a very nice model, and which I'm sure was correct. But then we basically made a prediction, and then yeah, it's interesting, but we had to wait for the crystal structure, or I mean, often they wanted to have it. They know the interactions in that. So then we had waited 3-4 years and they got a crystal structure that looks the same. So like it was correct, but it didn't really, I mean, they was not good enough to do phasing and anything like that, so they didn't have to get a structure, and they didn't really want to do any biology on it before they had a structure. We had one exception. It was actually happening a bit later, but we were using TRZ, or we use other methods also, and you can use any method to do it. And it was a protein. It was a CPA-AT transporter. So it's a family of proteins on CPAs. We had studied it for a long time, the evolution of it. And they are membrane proteins. And they're like, it's very interesting from evolutionary perspective because you have two groups or two folds. You have the ones that have like a helix that goes through it, which is broken, and the ones that have a reentrant region. So it goes in halfway through the membrane and out again. So one class of these that had these reentrant regions, there was a structure known, so we know, I mean, we had the structure and we could predict the structure well, but there was a subfamily that was truncated. So they lacked the second part of the second reentrant regions, because there are two reentrant regions that meet each other in the middle. So the second one was gone. So it was like, if you made a homogeneous model, it would have a big hole in the middle of the protein. I mean, it didn't make sense. It was like, but if you run this with TRZ or with another method, what happens is that you get one of the loops to become a new random regions. And it's a very, all methods give the same answer. And you have subfamilies and all things. But and then the other result there that you have one helix has to turn around and so on. So that, but I was really like, we were completely convinced that this model is correct. We still don't have a structure of it to experiment it, but it's like one of the few cases where you really could say that we can predict something new. We can predict something about it. It's not really, the model was not good enough to do, I would say, we could understand the transport mechanism and things like that, because it was transporting other, so it was not good enough, but really from the evolutionary perspective, we could learn something about it. But that was, I think, one of the few cases where we really could get some new biology out of it. So you had, so that was really a perspective. So that where you could do some volume. Yeah, good question here about how was the impact of using GPUs instead of CPUs. So GPUs was clearly one of the big reasons why image recognition improved so much. So the whole machine learning field completely changed by GPUs. And I mean, you can look at this image recognition contest, and it's the error rate drops from like 20 to 5% in 4 or 5 years. And that's and also that made that you use neural networks and no other methods, because that's perfect for calculations. GPUs, all other methods went out. So, and it also probably provided a lot of help for doing new algorithms and more efficient algorithms. So it certainly was a big impact. For prediction context, you would say that I think, I'm not sure that actually Raptor X Ultra Deep used GPUs. Don't think they did. TRSetta does. So you basically, but TRSetta, you probably can't, but it will be hard to train it on a CPU base, but it probably is possible still. But you had it. So basically, I would say that the jump between CAS 12 and 13, so it's like 4% GDTS to 6% GDTS, probably would be possible on CPUs, but very hard. You need a lot of big resources. But anything else that you need the GPU's power to calculate, because it's much easier. So it's just a more efficient way of calculations. Is that okay?
This is Rita speaking.
Yes.
In your experience, it is easier to predict a membrane protein or a globular protein? Because of the constraints, after all.
It doesn't really matter, I think. In most methods, you don't really see a problem. I mean, single spanning membrane proteins are difficult because none of these methods really take into account membranes. They don't have any physics. So you can easily end up with the extracellular and intercellular domain next to each other. And but in general, multi-domain proteins are more difficult. So it would be true for any multi-domain protein because the interaction between domains is less conserved. But in general, membrane proteins, I think it's not any difference at all. It works. Yeah. I mean, it's basically the co-evolution is the same type of signals, and so it's.
Because I was thinking in terms of the conservation overall of the structure, but okay.
Yeah, I mean, of course we have less data, but nowadays there's quite a lot of membrane protein data, so it's. Okay. So I don't think it's not a. I mean, whenever we try to look at it, it's not a big difference at least.
Good. Other questions around?
Then otherwise, I'll go on and go to the next part. Someone took now just typed something.
Okay, go ahead.
I'll go ahead, and if you have something, you have other chance to talk, so I'll go ahead. So enter the era of AlphaFold 2. So basically, everybody were sort of shocked at in 2020 December when we got the results for CAS 14. We saw the results, but I can skip this one. So basically, the results were amazing. So if you look at you all heard this many times. So you can look at here. This was the median GDTS, and basically jumped from like 60 to close to 90, and basically there's no significant difference between easy and hard targets anymore. So basically, all the targets are equally good. And 90 is probably pretty close to what you have on experimental accuracies. So if you take two different crystal forms, they are not going to be more than 90. And if you take away all other methods, basically they were as good as DeepMind was in CAS 13. So it was an improvement also, but this was I would say a big jump, particularly hard one. And suddenly, I mean, and it was also not like, otherwise often people look at small domains, but it was like all the structure was good. So my little talk, I'm certainly not the best expert in the world on AlphaFold, but there are a few. I mean, I think first one thing is really, if you look at the other measure, which is not GDTS, which is even more impressive, is this median RMSD error, C alpha. So this is how much error you have in median, which is 1 angstrom, which is even less than 1 angstrom. And all other methods are 3. So it really tells you have the accuracy that you don't have anywhere else with another method. So like, it's not that, the GDTS is sort of a good measure if you look at models that are 2-3 angstroms RMSD, then it's separate that for something that's going wrong. It's perfect, but it's not the best measure to separate something that is like 1 to 2 angstrom away. So this is, then RMSD is a good measure. So this really puts, I mean, you don't get this without getting all the side chains right. So you really look at the accuracies you had here. So I mean, look at the fold here. It's sure, every single loop is not perfect, but even like the benzo loops are just slightly off. And you basically, all the secondary structures are spot on here in green one. And the side chains here and around, this is, I guess, a sink, are perfect. I mean, every side chain is oriented around the sink. Right. The sink doesn't exist in the model, but it's sort of learned that there should be something there, should be sink, so they put the side chains right. And even the big proteins that gets basically right. It's not perfect, it's 2 angstroms RMSD, but like, except for like some small domain movement, it's almost spot on. So that is like, it was impressive. It's like, there's nothing, clearly these details was nothing else there close. Even homogeneous models are often not that good, unless they are very, very similar single entity. So you really got that. So what did they do? So there, I think there are a couple of things. I mean, if you look at their ablation studies, they really, it's not so clear there's one invention that is a big thing. There are a lot of small things that contribute. So one thing that is important is actually that they use this multiple sequence alignment as they put in the whole multiple sequence alignment. They don't took any measurements, calculations from it, as everybody has done before, where you take like PLMDCA or something like that to calculate from it, you do things and then you use that. You really take the MSA and use it full. They were not the first paper to do that. There was at least 2 papers earlier out that done that, but they probably did it in a better way than early people, and particularly that is through this Evo former, which I will talk about more in a second. So there is basically a way to get attention, which I know David mentioned. So you have attention both in the rows and the columns. So that's the key thing. And the other thing is that they really had these 2 tracks. So basically in parallel, basically jumping up and down, they predicted well, it's not the distance, it's the pair representation, which is sort of at least some information about the distance between every pair of residues and then MSA, and then jump back and forth. That is 2 tracks in parallel. Rosetta Fold has even 3 tracks in parallel, but and then you have this Evo format, you do this 48 rounds, and then you have a single, then you go back here, and then you put this into what they call structure module that actually builds all the modules here. And this is an innovative. If you actually, but if you skip that and just put it into Rosetta or something else, other model, it's not that much worse. It's almost as good, not really as good, but almost good. And then they're also very good at predicting what regions are good and what regions are bad. So high confidence and low confidence values. So there are a lot of engineering, a lot of like, by the ability to have a lot of compute power and a lot of good people around and a lot of like iterative engineering. You can add small progress in many small steps, and every progress adds up, and then at the end you actually have something that is much better than we started from. But then there are also, of course, a few key things like that has been around but not implemented altogether at once before. So this Evo format, basically it's a way to get attention between information on the sequence, multiple sequence alignment and the pairs. So you can basically look, the model can learn it, and it also has tricks like that, this triangular constraints. So basically, you use the distance between I and J and K, and then you have some constraints on the I and K distance and so on, and it does this in a very smart way. So this is like one block here. So it has like, basically you have this potential with pair biases, and then you go up here. And this is basically you do out the product mean, as always, and then you go back here, and you go up here again, and you have two representations. So you can iterate between them. They both go in and do both things. With the structure and the pair transfer. So this is a key thing, but it's parts of it has been existing before and things like that. There are the way, it's also a very smart way to do this structure model. One problem when you deal with 3D objects or even 2D objects in a neural network is that a rotation or translation of this object should give the same answer. So if you have a protein and you rotate it, it should have the same energy. Or if you calculate energy. So, and that is hard for, I mean, we think about it, we want to have Cartesian coordinates or X, Y, Z, but it's very hard for a machine to learn that. If you have like different rotations. So people have been trying around. There are ways to do this that are rotation invariant, but you want to have it both local and global invariant. And they sort of, I mean, one way, for instance, a simple way to do it is predicting the phi psi angles, or because that's rotation invariance. You predict that and not the continuity structure. But we know that that's not a very good measure, because like one small error, one error can propagate and become quite a big error at the end. So you're very, very sensitive to things that you could. Another way is predicting the context, as we did before, because that basically, and you then use something else to follow later. But here they predicted this, that is their structural representation or pairwise representation, and then they basically split. They ignore the chain constraints, the protein constraints. They basically say, in the beginning we treat every residue as a triangle in a gas phase. And then they have this way of doing this structural equivalence. They can just matter the order, but they put it in one thing and started and just build it up from there. And it doesn't matter which one, because it's invariant. And then they said, we're going to learn that residue I and J are going to be in contact. And of course, that's obvious because they're always in contact. So they're going to learn it quite easily. And then they just do this a few iterations of this one, and they got, even in the first iteration, you get a very good model that is, I mean, you don't have any, there are cases where you end up with broken chains. It happens, but it's very rare. That's when you have very little constraints. And then actually you can have this, you can even recycle it up to 3 times, 10 times, whatever. It seems like this recycling is only important when you have weak constraints. So if you have very, very strong constraints, the first iteration is fine. If you have very weak constraints, the recycling can help. So that's my short take on AlphaFold too. That hopefully provides some additional insights. I mean, there are people, there are a lot of YouTube videos out there talking.
May I interrupt you?
Yes.
1 second, just 1 second. So perhaps you can explain us a little bit. How do you take into consideration at the same time multiple sequence alignment and contact maps? This is really tricky for us, for me, for example, to understand the comment.
I mean, it's not the same. You see here, there are 2 tracks. So like, and then it's repeated many times, but like basically you have a multi-sequence representation and the parent. This you think you have whatever. I mean, you think this is the predicted distances. It's not exactly this, but it could be predicted distance between a pair, and this is the MSA representation. This is the MSA. The first step you do here is you take this predicted information and you have some kind of attention based here. So basically, it has, if you figure out, you know, of course, the position 1 here corresponds to position 1 here. Okay. So you take every row and every column and here and sort of train it somehow. I mean, it learns. I mean, it's added together. Every pair, for every column here, you have all the other pairs interactions, and then you do it. One run of rows.
You are telling me that this is the distance map, not the contact map.
It is not really. It's some distance map, is some hypervirtual space, whatever.
But and then the answer is the attention mechanism.
Yeah.
Okay. Very good.
And then at the end, you do some calculations here on the on these things, and then you basically have still it's like a one-dimensional. I mean, it's only a linear representation. You have each sequence with some MSA representation, and then you do outer product mean. So you basically go from one dimension to 2 dimensions.
Yes.
Okay. And then you do some calculations. That is updated triangles and self-attention, whatever, and you, on this triangle. And then at the end, you come out with a new MSA representation, a new pair representation, and this enters the next block. You have 48 of these blocks. So whatever this means in the middle, nobody understands, of course. But it's like but somehow they can learn. Fantastic. I mean, they iterate between them. Basically, it's not that they do things in parallel. They iterate between them.
So at the end of the day, during this procedure, they supervise whatsoever. Okay.
Yeah, but the supervision here, of course, is one of the things you can talk about. Is that the supervision, of course, is that it's all end to end. It starts over here with the sequence or MSA at least, and had a structure that. So certainly, of course, you can get the derivative of a change to the structure over here, all the back to change in one pyramid in block 24. So it's not trained step by step. It's trained the whole thing at once.
Yeah.
So whatever it means, I mean, it's thousands or at least hundreds of layers of networks that multiplication, but you can get derivatives all the way back. So it's like classical neural network training. I mean, a lot of tricks, of course, but like in principle, it's backpropagation.
Thank you very much.
So how good is it? So I mean, this is something we have a paper on the way out, or hopefully soon, together with a lot of other people, where you basically look what can you use it for. So basically, this is, I mean, very question if it's better or worse than memory protein. So this is basically the important part here is like it's very good at telling you when you're right, when you're wrong. So this is the residue predicted error and the real error. And you can see up here, you have most things have a predicted error and a real error, like 95% or better. But then you have a lot of things have done here where the error is worse, and it's basically worse. And this is using the LDT measure. So basically, this predicted error is very good at separating, telling you when you're right, when you're wrong. And you look at, if you take all proteins, you have about, if you look at PLDT over 90, you have about 50% of proteins. And that's compared to like only 40% of proteins where you have a template. If you go down to 70, which still is a quite good model, you have about 70% of proteins. And if you look at membrane proteins, plasma membrane proteins, I mean, it's not a big difference here. It's actually vesicles and organelle membranes are slightly better. Nucleic acid binding proteins look slightly worse for some reason. I don't know why, but I mean, in general, it's not a big difference between different types of proteins. And what is important here is like the ones that are yellow, below yellow, so less than 50%, this protein seems to correlate very well with disorder. So these are probably regions that are not ordered in structure. So basically, we will never get up to 100% unless we find a way of looking at disordered proteins. And you can also see that even the template-based iteration, I mean, template doesn't really matter. It's like you do almost as good without a template. So I never use templates because it's just one more step to do. So of course, people use this in many different ways. There are things like, what can you use it for? Like, I mean, you can learn a bit about this dynamics, this sort of flexibility, but it's not a perfect metaphor because it's really just predicting. You need to use additional information. It's really, maybe next slide is actually better. Yeah. So one thing is clear, it's very insensitive to point mutations. If you don't add any scan on a protein, it won't tell you anything. You get the same structure every time. And the scores will not tell you anything about what alanine is good and bad. I mean, you can be lucky though, but in general, it doesn't work. So if you want to know how important a model is, or a particular mutation is, you need to use something else. You can use fold X or whatever you want to use, but it doesn't work. It has no information. But the thing that might work is actually to truncate proteins. So maybe those circular mutations on it, maybe that could work. But so if you truncate something like in this case, talk about the CPA80 transporters, where you had part of the protein gone and it actually folds another way, that could maybe work. But I'm not sure it always works in the case. We had tried other cases, I'm not sure, but that could maybe work. People have tried to use different structural states. You have proteins that are flexible to different states. Doesn't work either. Sometimes if you're very lucky, you can get both states. And there are some techniques that particular Sergei Shlinikov, I think, introduced where you retrain AlphaFold and you might get it to work better, but I'm not sure that it's really well tested enough yet. But there are maybe some way to do it, but in default, it doesn't work. You get one state, and whatever you do, you'll always get the same state, unless you're lucky. Predicting the structure of large, flexible, repeated proteins, it doesn't work. So we have an example up in the right here is nebuline. So it happens to be one of my favorite proteins. So it's a very, very long protein with a short domain, which is 2 alpha helices. And you see, the predictions are not that bad on the local level. I mean, it's not super strong, but the helices are probably sort of predict. But it does not look like that at all. It's because it's actually binding to myosins, so it should be long and extended. AlphaFold 2 makes it a nice compact model. So that model, even if it's locally correct, is in the overall shape is completely wrong. If you take a single spanning memory protein, it doesn't know what memory is. So it doesn't. So in this case here, which is the protein next to it, it actually puts the transmembrane region next to the domain. So that doesn't work either. It doesn't know what the membrane is. It doesn't, and it would be very, very useful to have some way to put in constraints into the prediction. Say, I want these 2 residues to be separated by this amount. So some way to put in constraints, but there's no way to do that. That would be very useful. I guess there might be possibilities to do that somehow, but it's not done yet, at least. But that would be very useful, but you can't do that now. So that happens sometimes. We had some examples, some big DNA binding proteins. And of course, in reality, you should have had a DNA strain there, but of course, it doesn't know about it. So you just put things to be compact, so you don't get it correct. I mean, sometimes you get it correct, like in this zinc finger. The zinc is not there, but it puts it perfect anyway. But big like DNA, things that does not seem to work. So you can do it, but still we have this people on it, like you can try to understand structural space. It adds much more information. Not sure how. Still a lot of things missing. This disorder prediction is actually quite good. It's as good as state of the art, basically. So you basically anything that is red, so particularly low probability to be a low probability is disordered. And this is a protein here, and so the fragments here are correct, but the disorder is wrong. So it's one of the big uses is to use to solve structures. So people doing, I mean, there's this very nice paper by Jon Kosinski about the nuclear pore complex, where they really had a big help of adding models from AlphaFold and also from RosettaFold to model low resolution cryo-EM data. So for that, it's extremely useful. It really helps to solve structures. And I have my colleagues have probably 5 or 10 structures where they couldn't face the crystals before, and they had put in an AlphaFold model and a kind of molecular replacement. So that's not really. You can do things like predicting variance or things like that if you use it as a model. And the model is often, if you have a good model, it's as good as the crystal structure. So any method to predict variance effects, you can use this crystal structure. And anything where we basically could use a structure, we can use an AlphaFold model. So any structure, any bioinformatics method we do. You probably can use it for drug design in some cases, but it's not super easy because you have a lot of faults, a lot of, particularly disordered regions you have to deal with. So but you have proteins without them, and you could, in some cases, get a good idea of the binding pockets. Okay, I thought of having, we still have time, right? So I guess we could take some questions again. So there was a question about these flexible regions. We have looked, so basically the idea about flexible regions is that at least one idea is that some of them are folding up to a well-formed structure when they bind to something else. That's at least one idea people have. So we tried to do that, look into that. So we took the displot database, and there are annotations there of regions that fold up on binding and do not fold up on binding. And clearly, there are methods. Clearly, we thought we could basically see the ones that fold up on binding. We could get better models for than the ones that do not, but we couldn't see any pattern for that. So even it's very hard to say if two proteins. If really they are folding when you bind something, or if it's just that we don't have a signal for it, not like that. So, and this alternative phases, alternative conformations of flexibility seems to be very hard to get to. So at the moment, I don't think we can do it. Yes, no, the next question about constraints, how you can put it in. I think it's possible, but I think you need to retrain AlphaFold. You need to redesign it. So there are probably few people in the world that can do it. There is an open fold copy, basically, you can copy the project. So in principle, if there was some very good computer scientist students that really want to spend some time looking into the code and trying to do that and think about it, I could imagine that you could do it, but then you probably might need to retrain it somehow. So it's not obvious. It's not obvious how to do it, but and it's not like, because everything is end-to-end, everything is integrated. It's quite hard, but it would be. You sort of could retrain it on anything in PDB with a large ligand with DNA or something like that, and you basically put in some constraints from that in some regular way, and you retrain everything with these sort of constraints. You should have it's almost like putting the template extra, but in some other template, and you try to retrain it with these constraints. That probably, I think that could work, but I think it needs to be retrained, and at the moment, it's only DeepMind are able to train it at all. So they would need to do it at the moment, but I'm sure it will happen sooner or later. But yeah. Okay. So anything else about AlphaFold? So. No, it's clear. It's like, of course, a couple of other nice surprises was that it was actually easy. They made it easily available. Yeah, I think it was a big concern like a year ago, when people thought, they're going to make it, nobody else can use it. It's going to be, they told you that it took days for them to run a model. And that's, I don't know if they had a slower implementation, if there was other problems, or they run it in a longer way, but at the moment it takes, the folding can take minutes or an hour maximum. It's like, unless it's something very big. So it's really, that they made it available in a good way is very nice of them, I would say. And it contributes to them also. And that was probably the right way to do it. Okay, anything else? If not, I'll go on. Oh, that was just on whatever. So there was a question about that Alex Bateman showed that PLT is probably overestimated for short proteins. Did you account for that when connecting disorder proteins? The disorder protein is not my part. I haven't done it. The answer is I don't know. I'm sure that PLT is not the perfect measure. And talk to Silvio Tosatti and people that do this, they say there are other methods that are better. It's not perfect disorder predictor, but it's not that bad. It's quite close to state of the art. It depends on how you measure, what you measure on. And at least it's a useful tool. But disorder is an important problem. We'll come back to it in a second. That is, they have to consider what you do with it in proteins, because they are in the AlphaFold. So we'll come back to that in a second. Because I don't think even they know how to deal with it in the best way. So back to our work on protein-protein interactions. So I also had to jump back to basic Cas13 or Cas before then, and I go on until things done last week. I think I was doing it in 2 parts also. So somehow it was obvious for many people in the field that the next big challenge is not protein structure prediction, but it's to predict interactions. And we know that interactions are very, very important for all biological functions. I mean, and also the interactions are very different types of interactions exist. You have like big molecular complexes. You can think about the ribosome. It's big machinery, maybe not completely static, but it's like more or less the same subunits are there all the time. I mean, some regulatory things. But you also have very transient interactions. You have something binding, something regulating for a small second, and you have all these slims, so these short linear motifs that are disordered and bind things and that such. You have a very big variation of interactions. And there's also been a lot of experimental studies on interactions for the last decades, of people trying to have experimental methods to interact, tell you what two proteins interact and how they interact. So interactions are important, clearly, and we're still missing lots of information from it. So often you have interaction network that is dedicated to that, and it's kind of, people have studied this network in many years, and we have done it, other people have done it, and it's like you have this interaction hub. So some proteins have many partners, and most people only have a few, and there are different types of interactions and things like that. So there's a lot of work there. The question was, can we get from this multiple sequence alignment somehow to interactions? And the way we talk about it is basically we do it by structure. But people have done it without structure. So you can basically take 2 MSAs, have some smart machine learning method, and ask does this proteins interact or not. There is a couple of papers about Bonner-Berger, for instance, that has done that with very impressive results. I am not convinced that they are trained in the right way. I think they're overtrained. So I'm not convinced that they really hold in large scales, but there certainly is work going on in this area. So it could be, if you find the right way to train it, it's not unlikely that you actually can predict these networks, ignoring structure completely, to some extent at least. So what is the problem with interaction? So the problem here is partly because we don't know what proteins interact. So if you think about, you have 2 proteins. So in this case, you have 2 MSAs. They're not the same, but I just copied it, right? But each row in this MSA represents 1 protein in 1 species. So you have a yeast protein, you have a human protein, etc. And the yeast protein will never interact with human protein in normal circumstances. So you would like to pair, if you want to calculate this correlated mutations between proteins, you would like to pair the rows here so that you have the ones that are interacting. So the human protein is paired up with the human protein and the yeast with the yeast. So that sounds quite simple, but it's only simple if you only have 1 copy of both proteins in each organism, which is not the case. I mean, in bacteria, it can happen sometimes, but but in most cases, you have a lot of paralogs, you have a lot of different copies that are homologs to each other, but are quite similar. So you assume that you have assume you have a protein that is in one species, they interact, and then you have 4 paralogs in each species. So you have five copies of each protein in all species, in one species. Then you have to figure out which of these 5 interact with other 5. I mean, and it could even be so that 1 interacts with all 5 in different circumstances, or they interact exactly pairwise, or it's not trivial to figure out exactly what interaction patterns happen there. And if you do it wrong, you get noise in your correlations, and your mutual information signal is lost, or coevolution signal is lost. So that makes it quite hard, and particularly for methods that in many sequences, and there are ways to do it that are hard. But you can try, and often the easiest way to try is basically you start with 1 pair of sequences, then you want to take the top hit in each genome. So if you have paralogs, you ignore them. But there are other people that have done other methods also that try to evaluate it and iterate it and do some optimization process, but that's very slow. But in our case, we used to use top hits, and that's basically what the AlphaFold tool also doing later. But there are probably better ways to do it. But this pairing is a big origin of the problem. It could be another problem also. The difference that interactions between proteins is probably less conserved than interactions within the protein. Because we know proteins can interact with many proteins and they can evolve quite fast. They might even interact in different ways. You have homomers that can be in different forms. There can be dimers or tetramers and so on. So there are a lot of things that are not obvious. I mean, think about myoglobin and hemoglobin. Hemoglobin is tetramer, myoglobin is a monomer. The proteins are clearly homologous, but they interact completely different. So anyhow, you can do it. So you can take like methods, try to pair them. And in that case, what we did here is basically you can just fuse them, you put some polyglycine in between, and you run your favorite program. In this case, you run Tia Rosetta. And you have in the upper right hand here is the correct contact. So you see you should have contact between let's do 50 and 400. So 400 is the second chain. You should have some contact pattern here. And you try to predict it in the bottom part. So you can see in this case, both cases, this protein 1 and protein 2 are well predicted. The contact maps are very similar, but the interaction pattern is wrong. And this results in that we have the right fold, and then we can put this into the Tia Rosetta and fold and dock two proteins at the same time. But you see in this case, the proteins are not... each protein itself is quite well folded. It's not perfect, but it's quite good. But interaction is completely wrong. We put it in the wrong place. I mean, it's sort of the same vicinity, but it's clearly wrong in the docking. I mean, the blue one and the dark blue one and light blue one are not in the same place. So in this case, it didn't work. But anyway, we have a paper out where we did this, and we did using Tia Rosetta. So we published this, I think, it was done basically for CASP 14. So we had it running then, but I think it was published last year. Since bioinformatics. So we do this, we do alignments, we do these merge alignments. We just run Tia Rosetta, basically we modify the pipeline slightly, but minor things. And we could follow dock this. So in some cases, we could actually get some kind of nice models. This looks not too bad. Or in particular, and the blue and dark one. And then actually we also developed a scoring function that was basically based on consistency. So basically, if you run it 5 times and you get the same model every time, it's correct. If you get different models, it's most likely wrong. So that was the best scoring function we could find. So classical consensus scoring function. We call it pconstock. So we did that, and at least in some examples, it's nice. We are very happy to get these predictions. It's really, the contact map is not maybe perfect, but it's quite good. It's maybe 8% correct. But you see, the model is correct, the folding lock is correct. But if you look at it overall, in general, this is usually called docu score, where like 0.2, 0.23 is often cut off for good and bad models. It's a handful. It's like 15 or 8 to 10 models that are good. So it's really out of 200 or something like that, 220, I think that. So it's really very small parts where we manage, but it's complementary to other methods. So we can compare it to methods, classical document methods, or even template-based document methods. It's complementary, so at least it adds some value, but it was not. Most cases we failed, which is most cases are close to 0, which is sad. At least in CASP 14, we had one model, but we did the best prediction of our interactions. This also like in CASP 13 or CASP 12, we had one good prediction, and it was also a bit like. Actually, the best prediction was a bit a combination. We used this model, and then we used some template-based modeling and picked out some examples where it was that looked the same and used that one. That was our best prediction. But that prediction was far ahead of anyone else for this pair of interactions. And it's not, actually, I don't know the answer because I haven't looked. I don't know. So this is our model just compared to another model. So At least prediction looked good in one case. But obviously, we and everybody else, or not everybody, many other people, realized we can try to do the same thing using AlphaFold. Exactly the same idea. We make two alignments, we sort of merge them in some way, and we put it into AlphaFold. So instead of using zero set, we use AlphaFold. And in the beginning, people actually put this linker here, but you after you realize how you run it, you don't even need to do that. You just put a residue number. You just, this is residue 201, and then you take this run rest, you add 200, residue 400. So it treats it as a long chain with a big gap, because AlphaFold can handle gaps. So we did that. I will come back to exactly what we did later. And this is the docu score of the methods we used on the benchmark set. And you see the Rosetta Fold was had like 10% correct. Graham in this case is a documented had like 20% correct. And so there it was a Rosetta Fold. Our Picos doc probably also something like 20%. AlphaFold 2 without an optimization had 44%, and then we sort of combined the alignment in better and better ways, got up to 59%. So that's a quite big jump from like 10% to 60%. And clearly, I mean, so we were happy about it. So one trick we did was exactly this alignment. So like you can think about, you have, all right, there's three ways to do MSAs. You can just put in the merge sequence into H splits and run alignment. And actually, sometimes you get things that span both sequences A and B because they're overlapping, because they are merged proteins in BDB or in unipods. So you get alignment look at that. So that was the default one that we got 45% accuracy in it. You can do paired alignment where you do exactly what we did before. You try to find the matching species and do it. Or surprisingly enough, you can also do a blocked alignment. So basically just put alignment aligned to gaps. And even the blocked alignment, I think, is not, well, actually I don't have here. Well, the blocked alignment helps something. So if you take the paired alone, you get 49%. With the paired, if you put both these alignments together, with 58. So in some cases, the blocked alignment actually works. You work with the blocked alignment alone, which is just contributed to AlphaFold 2 to be really good at recognizing patterns, because you have no coevolution in that case. And then we have some way to do racking. And then later, AlphaFold 2, DeepMind has released something called AlphaFold Multimer, which is a method that can do exactly this, but is also retrained for multimers. And in this case, it does slightly better, but it has some other problems I'll mention in a second. So one thing we looked at, if you looked at, does coevolution work? So basically, if we run a DCA method and say, do you have interchain contacts or not? So you can divide them into ones that have no contacts and the ones that have many contacts. You can see if you use this paired block alignments, we get better performance on the ones that have contacts. So if there is a DCA signal, AlphaFold uses it. But there are cases where there is no signal and we still do good alignment. But there are fewer cases and still maybe helps these cases. But it really, if you have a coevolution signal, it works. It adds information. So this is the accuracy. If you look at the side chains, it's really, really good prediction. Even side chain packets here are often correct. And you see some loops are not 100%, but basically, when you get a nice model, it's close to experimental accuracies. We also want to ask the question if you could separate good and bad models. So we had that, because AlphaFold could not do that because the scores are not very good. But we just took two of the numbers. It looked like basically the size of the interface, so the number of contacts in the interface. And we took the average accuracy or predicted accuracy in the interface, the interface PLDT, predicted LDT values there. And then if you plot this against DocQ, you see that most of the good models have many contacts, about 100 contacts, and a good PLDT. So we can just fit the curve. So we take the average, this number here, multiplied by the log of that number, because you have a log scale here, and you get a nice sigmoidal curve. I mean, it's not perfect, but it's pretty good, called PDocQ. It's a predicted DocQ value. So it's not perfect, but it's pretty good at predicting DocQ values. So that gives us a handle on separating correct and incorrect prediction. It's actually pretty good at separating interacting and non-interacting proteins also. So you have a ROC curve here separating good bad models. You have like a ROC curve of 0.95. If you're in the benchmark of binding non-binding proteins, you have a AUC of 0.87. So it's quite good. Which is useful for what we do next. So some examples. This is an example. We have a bad model where basically should be RNA binding. We got it wrong. So this RNA DNA here should be, it's not there, so the model is bad. But you have some good predictions here also. Even big complexes are predict quite well. And in this case, there was the few new structures that were not used for training. So they are really made after AlphaFold was released. So that at least works in some cases. So it's not completely overtrained. And we can see that basically it works for all types of proteins. It's a bit better for bacterial protein. It's a bit better if you have the interface that are, I don't know, it's not a big difference, but you have maybe a bit better if you have sheets in the interface. It's a bit better for bacteria. And if you have more sequences, it's maybe slightly better on average. But in general, it works quite often, but not always. But bacteria is better than human proteins. So next, now we have this measure. And now we can take experimental data sets of protein interactions. We took two of these data sets. URI, it's a data set of yeast-2 hybrid studies of 17,000 human proteins. So 17,000 pairs of proteins. No, 17,000 square number of pairs. So it's a lot of experiments, and they've done everything 10 times. So they have 64,000 interactions plus some benchmarking. So they have like 65,000 interactions, something like that, involving 50,000 proteins. And another data set is the UMAP2, which is basic purification data, where they have basically taken the human proteome, purified it in many different ways, and whenever two things always end up in the same purification batch, they assume that they interact, and then they make modules out of that. So then we took the top 11,000 proteins from that or something like that. And then we can basically run these and look at the p.q values for these. So we run all these pairs, and if you see the red curves here, basically first, that's basically things where we know the structure. So it's a basic data sets where we know they interact. So they are either from PDB or from cross-linking study or something like that. If you take a random set of proteins, you basically have a very, very few things that have high p.q values. The random sets are very good there. But if you take UMAP, you see you have like two peaks. You have like one peak here of things that have very bad values and one smaller peak with things that have good values. So at least a fraction of the UMAP values are predicted to interact. The URI is even much less so. This is very, very small peak up here, but there are something, but it's a big data set also. But clearly, there is a subset where we predict the interaction to be good. So we have focused on that. So this is the same plot again. You can see the numbers down here, how many are in each set. And this is just some examples of good predicted human proteins. This is human proteins only. And some benchmarks for PDB, and the predictions are good. In some cases, actually, prediction is good even if the p.q is bad. So there are cases down here where good predictions, but we can't really separate it because they are small interfaces and they're probably part of bigger complexes. So there are a number of good predictions that are good. You can look at them. And we have in total something like 3,000 predicted complex pairs. And you can see here basically, well, this was another cutoff. So it's basically a larger patch of URI, of UMAP and of URI. Yeah, so here, well, it's the same again, more or less. So we can do things, we can look at cross-linking data. So we have a case with cross-linking data, and of course we can see that it's enhanced in cross-linking data. So there are a number of, this is a novel prediction that doesn't exist, but we have also cross-linking data, or not we, but our collaborators, or Peter Beltro's collaborators, have cross-linking data that can link to things that should actually interact, and our models agree with the data in about 80% of the cases on that. It's not always perfect, but even things that do not look, they kind of extended, we have nice predictions there. What's interesting, if you start looking at this content in this network map, so you look at, basically we have a network map of all interactions that are from experiments, and then we also have a map of looking at how things are predicted. So like, and you can do nice maps of this one. So you can look at, I think, two examples of this map. So you look at this map up here on top in the beginning, you have one center protein called P31150 interacting with like 10 other proteins. But these proteins are not interacting with each other. There's no data for that. And the reason is, of course, if you make a model of it, you basically see that they all overlap. So you have one green protein interacting with all the other, and they all interact the same way. So basically, that means that this is a protein that has many different partners. It's been binding 10 different partners, by all homologous, but they all basically bind 10 different partners in the same way. So they're competing for the binding of this protein. And that's contrast to what you see the network down here, which is the proteasome, where everything is interacting with everything instead. And we also have good prediction of everything. So you really can get information of details of how networks of interaction occur. It's not just say, oh, they interact. It's also like they're interacting or they're competing with each other or they're all interacting with each other together. It's a big part of a big complex. Yeah, and the structure really helps there to figure out what's happening there. Yeah, so I come back to what we use applicable this proteasome in a second, but I will, and then I just tell you about this AlphaFold multimer that is benchmarked, and it's pretty good for dimers and trimers, but it force drops the bigger complexes we have. And it has a big problem. It doesn't handle disordered proteins, disordered regions very well. So if you have disordered regions in your protein, it just overlaps them. So it gets messy. So it's a bit of a problem, but it's in general it's better than our pipeline, but it's not. But it makes a lot of overlap. So again, I think still we have time for a short questions, and then I'll come back to the last part of that. I see there's something in the chat. So this disorder regions are handled very different by AlphaFold multimer and AlphaFold original AlphaFold. In AlphaFold original one, as you've seen, basically they put them as long strings putting out and over. They are flowing around and being very, very long strings. In the AlphaFold multimer, I guess it's trained to have everything very compact. They end up to be in the middle of the protein and overlapping. So they are not, none of the predictions are correct, but these things that have everything in the middle makes it very messy. So it doesn't make physical sense because they're really overlapping. So that is the problem with the disordered regions in AlphaFold multimer. So we have dealt with it in some cases where sometimes you cut away the disordered region, and that also makes it better. There may be ways you can ignore something, but DeepMind has said, they told me already 2 months ago that they're going to make a new version that works better, but it's not released yet. So I think there's something in the training procedure that really makes it happen, and it's probably not that easy to fix. Because John Jumper told me in December that they should do it, and they haven't done it yet. Okay, I see new questions. Naive question: could you suggest a method tool to carry out protein docking by beginners with access of resources to carry out molecular dynamics? We don't do any molecular dynamics. If you just go to our GitLab repository, you can just download and run it. So basically, you can do it with the AlphaFold. So if you want to do, I guess you might have heard about AlphaFold. I can show the screen in a second. So yeah, there is this fantastic tool developed by Martin Steinegger and Sergei Shinnikov, mainly. That is, I skipped over that slide, didn't talk about it, but it's not there. So if you go to this AlphaFold, you can Google for it. And AlphaFold is, if you go to advanced options here, you can actually do docking on that and AlphaFold multimer. You just click on it. And AlphaFold or Colab is basically a way to run any code you want on one of Google's clusters for free for up to 12 hours of that. So you can use it for some time. I mean, I'm sure you can pay to get more, but that's one easy way to do it. Otherwise, you can download our code or AlphaFold code to run it. It's quite easy. But if you don't find it, send me an email. And to run it, basically what you need is a good GPU. And the important factor is sort of, for small the size of the RAM on the GPU is what is limiting. It's not really how old it is. So if you only have like 2070, 2080 with 8 gigs RAM, you can only run like proteins of a few hundred residues. We have things with 40 gigs RAM, and we can run things up to 3,000 residues. So the size of the protein is really RAM limited on GPU. And so that might be a limit for most people, because a GPU with 48 gigs of memory costs 6,000 euros on that. So that's a bit expensive, but that's basically more is more. Yeah. As an NMR spectroscopist who studies disordered regions of proteins, how can we optimize the data, the positions which can be used for future AI methods? Predict. I think, how do we train? It's a very interesting question. We know that dynamics is one of the big challenges. We want to do better dynamics. And I think it should be possible to use this type of method to predict dynamics if you had something to train it on. And not being a complete NMR expert, but knowing something about it, I think the way that most people try to do is wrong. I think people are trying to basically look at the conformation ensembles to look at and try to predict that. And that is certainly very dependent on how many constraints you have in NMR, what temperature you do the constraints on, but also on your optimization protocols. How do you do, what distance geometry do you use, and so on. So it's certainly not an unbiased sample. So I think what you would like to have is the real experimental data on dynamics, like hydrogen exchange or J-couplings analysis, so on, which I'm sure you know much better than I. But if you could have this as much detailed experimental information about dynamics, how it should be implemented, I don't know, for as much detail as possible. But if you really have, even just basically something like B factors, flexibility, RMSD, certainty, something that would probably be useful. But the difficulty is that it's such a range of things. So it really is completely disordered to just some vibrations. It's hard, but yeah. So yeah, I said problem perception regarding. There was a question here about. I referred to the Berger paper, the descript paper. I think at least I can't say that. The problem, I think, what is the problem with that paper? I think the problem is that it's not cross-validated correctly. I think what they have done, if I remember correctly, is that they have cross-validated. They basically have very different positive and negative sets. So, and they cross-validated. So I think what you need to do, which I haven't done, is that you need to basically have the same proteins in the positive and the negative set, but just interacting in non-interacting pairs. Because now what they can do is basically can learn properties of the interacting pairs that are not directly doing interactions, but are just properties of these proteins that are different from properties of the non-interacting pairs. So I think if you run this, when we try to train it, we can also get good performance. But if we trained it with negative pairs that were random, say the same proteins, but just randomized it, assuming that two random proteins not interact, we could not train it anymore. So that's but I haven't done it carefully enough. It might be something else, but that's from our very quick try to do the same thing. That's what we found. Okay, any more questions? Otherwise, I'll go to our unpublished data. So that could be interesting also. I need to go back to where I was.
Just to sum up a little bit, may I? This is Rita speaking.
Go ahead.
To sum up a little bit, so AlphaFold 2, did they release a version for protein-protein interaction? Yes or no? This is unclear to me.
Yes.
They did.
But the paper is in bioRxiv. It's not published yet, but it's in bioRxiv.
Okay, so it's the paper in bioRxiv. Okay, that I know, because otherwise I... No,
the paper, the code is available, but the paper is, at least it's not out yet. So that's, and I think my guess is that is because of the problems with these disordered regions.
Okay.
So that's my guess.
Then your paper, taking advantage of AlphaFold, but including also your developments, that is out.
It is accepted in Nature Communication. It's not published yet, but it's accepted.
Reason why I'm writing a review right now. So,
so the paper, the bioRxiv version is there, and the other one is in Nature. The paper, this complex paper is still under review. I mean, it was rejected from one journal, and now it's submitted the next one.
Okay.
That's not out yet, but it's a bioRxiv also.
So, any other suggestion on the fields that you are using within framed into the docking procedures?
I will tell you now. In the next
okay, sorry.
So this is what we know. I will. So what we found interesting was this idea of building complexes. So we wanted to build. I mean, we thought it was like we from seeing these networks, we thought these networks were interesting, and we realized we could maybe try to build complexes. And I mean, so we just looked at some here, and they mapped our predictions to it, and it looks nice in some cases not. Like, and the network is different. So we wanted to build complexes. So in this paper that is still under review, we did in some cases, we put in some data here. In some cases, we can build nicely. We did it manually. We basically took like dimers and put them on top of each other, and I mean, you have a dimer. You have A and B, and you have B and C. You superpose the B chains on the top of it, and you have A, B, C. And we did it manually. So like we took one and added the other one. I did some automatic script that worked somehow, but like we did it a bit like manually. And we have some examples that we could build things, at least parts of things that looks nice. But we sort of know the answer, so it's not really cheating a bit. But so the favorite example is part of the proteasome, which also highlights some of the problem. So the proteasome has, I mean, the 20S is just the bottom ring here. It has 14 different chains, but 2 copies, it has 28 chains. But they're all homologous. They all look the same. So they are basically all have the same structure, but they are not different. So one problem we have is we can't, I mean, if we run every pair, everything runs with everything, and we get the same structure every time, basically. So we can't separate the interactions from chain A, I mean, which pairs are really involved, because they look the same, and the methods are looking exactly what interacts and what does not. We cannot do, but we could build the whole ring here quite accurately. It's not perfect, but we could build the whole thing manually from pairwise interactions. But the chains are in the wrong place, but they are very similar, so the structure is the same. So that we could do manually. And basically, you think about the procedure here. You start with all the interactions, and you add things one by one. And you can think about it like an interaction network with everything interacting, and you keep on building, and you're adding one more chain at a time by superposing them. And the only thing you do is you check that it doesn't overlap with anything else, because then you throw it away. So you can do that. But you see, you have a network like this. So this is really, I have to acknowledge this is work by my student, Patrick Bryant, has done it completely himself. So I shouldn't take any credit for it. But you see, you can build things, but there are also many different paths. You can start with one path and start another path. There are many different options to it, and some work, some do not. That's the problem. So, and you see, this networks looks very different in different proteins. These are some examples. Some are like a circular ring, and then it's quite easy. You have to add one to another. Some has one big component in the middle, terminal cells. This is a proteasome. Some are like just separate from each other, and so on. So there are different networks, but they are in these interactive networks are different types. But in principle, you can do this. It's like you have a pipeline, you start at node A, and you maybe have AB and you want to add C to it. You take the BA dimer and the AC dimer, and you try to overlap them. And basically, Chef does C and B in overlap or not. If you don't overlap, you keep it. If it overlaps, you stop. Let's try something else. And then that's some scoring at the end. So he started doing that. In the first step, he assumes that he knows what interacts with what. So basically, you can assume that you have, you know the interaction map, so this sort of graph. And then, but still, it's still a lot of options. You can start forever in one place, and you can adding things in random order. So you can easily end up with tens of thousands of different paths through this graph. There are a lot of them. But many of them won't work because things start colliding, so you can start building here. So at least in this case, it worked nicely. And and we can use a scoring, so we can basically use this P.Q score that we have, or a version of that, and just sum up overall interfaces, and we get a score. And then, so if you can assemble different ways, we take the one with the best score. So that's quite straightforward. But one problem is like in case one problem was that they have this overlapping predictions. This is like predictions by AlphaFold multimer, you see here, and they are basically on top of each other. So everything's overlapping. And of course, if they're already overlapping before we try to merge together, after they're going to be overlapping also, so then our pipeline going to sort them out. So we had to go back to our old pipeline for most cases to ignore that. Another problem is like if you start with dimers, you can't manage everything because like you always get the same interactions. You don't get all the interactions. So we had, because like this purple one here that should be done here, I guess, is end up in the middle, end up in the wrong way because all the dimers, you don't get anything. So we had to include trimers also. So we had to model all the trimers also, which is a problem sometimes because it sometimes crashes and they're too big, but in general, most cases we could. So we basically do dimers and trimers. And then in this case, here actually, this upper row here is modeled with nodal structure. And the bottom one, we assume we don't know the interaction map, but then we just try all possible ways. And you see that we can see some big structures quite accurate. So the team scores at 0.9 in many cases. I mean, you see here, this is not everything, but like 1/3 of the models have like 0.8 or higher TM scores. And like half the models have 0.6 right here. But 0.6 is not that good because like it means basically half the model can be wrong. So like it's not that good. But in this case, sometimes it's not that good, but it still is like one part is correct. So like we have one part in the middle is correct, but we don't get the outer ring, we got the inner ring here. So we basically, for about half the protons, we can get a model that looks quite good. And it's up to 20 chains, something that we can do it with. I mean, there are some success rates, like these are, so it's about, yeah, successful in these cases, like for homomers, it's like 25%, for heteromers, it's like 10% in these cases. It's better for bacteria than for viruses, and viruses than for eukaryotes. Viruses there's only 6 of them, but they are all successful. Yeah, I don't think they're so important, but basically they are better. We can compare it to AlphaFold multimer for hexamers, and basically there are some cases where the AlphaFold multimer is better for hexamers, and some cases were worse. But I mean, they are in general about they're kind of comparable. So there are cases where AlphaFold multimer is better and where we are better, but AlphaFold multimer cannot handle 10, 20 chains because it's just too big for that. It doesn't work, it crashes. So we can't even run it. So we can't compare the biggest things. So this is one approach to try to make bigger things by using these sub-assemblies. Yeah, so this was the last part of that part. And remember, this is not published yet, but hopefully we'll put it in the bioarchive next week or something like that. And yeah, this is how you can run it basically. AlphaFold, this Colab I talked about. If you have a big computer, you can run it on. That's what we do. There is, of course, I guess you heard about the AlphaFold database. Uniprot has, I don't know how many millions of structures there already. You're going to have basically everything at the end of the year. And of course, if you have a big laptop, you can run it also. So and it's not that. I mean, if you have a GPU, it's not that time consuming. I mean, running MD takes much more time, but you need a GPU. So it takes a few minutes, or maybe 15 minutes. I mean, it takes longer time to generate the MSA unless you use MSX2. But it's like the MSA generation is often a bottleneck. So it's not that bad. Had to thank the people. Patrick and Gabriella has done the development here, and Julie and Aditi has done a lot of the benchmarking. The interaction map is together with Pedro and his people. And Petras is involved in a lot of the prudent docking here also. And some people that pay me money or give computer time. So that leaves us a bit of time for the final questions.
Thank you. There are questions. You can raise your hand or open your microphone.
There's one thing in the chat.
Yes, there is
one. Recent publications suggest AlphaFold does not as well when predicting the impact of segmentations. Should one then? Yeah. So yeah, I think I said it doesn't work at all. I would. People do it, and I think it's wrong. I mean, if you want to have a predictor mutation or a predictor of something small changes, it doesn't work. So what you need to do in that case is to use something else. If you want to predict, I mean, if you want to have a single point mutation, you should. If you use AlphaFold to generate a model, and then use Fold X or Rosetta or whatever standard tools you want to use for predicting the effective mutation, but you cannot use the AlphaFold to generate different models. That doesn't work. Okay, and other questions?
Okay, I have another question myself, but I don't want to interfere with the students. So my question would be the following. At this stage of protein-protein interaction, are we dealing with a sort of, I mean, we are trying practically to build models in which where we already know that they are interacting. So to say, we have PDBs in the databases in which we know that complexes are formed, even crystallographical, in a crystallographical way, in an MR way, whatsoever. Then we have all these interaction maps around, and we know better than you know better than I do, possibly that all these interaction maps, they have very little in common sometimes when you are trying to compare them and to look for all the different protein that eventually interacts, you find very little that is conserved from one map to the other one. So they are changeable. Now the question, with this type of approaches, you are, or people are, trying to model what is possible to model within the interaction maps, or are they finding or searching for new putative interactions?
I think the methods were, I think the two things. First, we have to start distinguishing different types of interactions. So what these methods can do is that I think they can model stable interactions in complexes. That are things that are binding. We have an evolutionary signal that things are binding quite strongly to each other. The methods do not work for transient interactions, so like short-time regulation. So the real things that are, with exceptions maybe, but in general, I don't think they work. So the first thing we probably should do is to separate interactions into at least two types. The second thing is that the methods works extremely well at separating this type of stable interactions from non-interacting proteins, from non-stable interactions. The problem is that that's a lot. If you want to run it, it's purely computational. And you can do it for yeast to E. coli, because you have, at least with some, you have 25 million pairs. At least that's possible to run. It's a bit pushing it. We run AlphaFold and everything, but with some tricks, maybe you could do it if you do some clustering or something. But the human is 400 million pairs. So computationally, there's no way we are close to do that.
Okay.
So, but in principle, it should work. So you probably, so the main reason to use these interaction maps is just to increase the number of potential hits. It's not because they are very useful in another way, but they at least gives you a smaller search space.
Good. Since, in your method, you are including doc, whatever it means, because I'm not aware of all the details. So basically, when you have a docking method, you are trying with some sort of fields, and you do practically molecular dynamics, and then you are trying to search for putative new interactions. So I was wondering how the two things are, joined one to the other one. So is docking sort of providing more details and more, I would say, correct interactions, or filter out whatever the AlphaFold is?
So we are not doing docking in traditional way. It's just a word. So we treat this as one system. So it's folding and docking at the same time. So it has nothing to do with, it has nothing to do with docking in traditional way. And as you know as well as I, protein-protein docking, there are two types of protein-protein docking. They have the ones that is called template-based docking, which is basically homology modeling. That of course works if the template is good. But the ones that are shape complementarity, put things together, it has never worked, to be honest. It's there are a lot of assumptions. In a few cases it works, but like in general, it's a mess. So that's kind of useless, I think. And this is much more powerful here. So that method, I think, is dead.
Okay, fantastic. No, because you know the problem here. In the PDB, we have whatsoever it's stable interactions, or I don't know, proteins that eventually are found stable during the crystallization process or whatsoever. In reality, we have many proteins that eventually comes together, and then they do interact as long as they have to function together to do something. So this scenario, which is, I believe, the real scenario in which you have strong dynamical components in describing whatever is protein-protein interaction. Is this feasible nowadays to model, or still we have the problem of, I don't know, finding new partners?
I think we are... we are starting to see this problem when we run real cases. You start seeing that you have long disorder regions that mess up things, and they are certainly things that are. I think we can only. AlphaFold has only learned what is in PDB, so you can only do PDB. So basically. It was lucky that you could extend it from a single chain to multiple chains, because it sort of, it was the same method, it sort of worked. It had to train on that, but the physics is the same. But to get information about things that are not in PDB, it's not trained to do that. It doesn't learn physics. So I don't think that's realistic at the moment. You would need to do something else. You would need to train an energy function that is sort of more general or something like that. It's like you need another approach for that. So I don't think you can expect to do that.
Okay.
There was a question in the chat there. So they asked why AlphaFold 2 was predicting the predicted aligned error, and that was supposed to be shown to be indicative of PPIs. So the reason why we didn't include this was we didn't try. So that's we actually, I think, we basically never used it. So that would have been a good idea to include that in our PDoc score. One practical reason is that it's just that the PLC scores is saved in the PDB file, so it's very easy to get access to it. Otherwise, you have to save the whole pickle file that it has that you need to save and take hours. It was just practically a bit so harder, but it might be a good idea. People should have tried if we haven't done it.
More questions? Students of the master, please.
Master students, interesting.
There's still people here, haven't left.
No.
No. You have still 140. Oh, but perhaps you can see it yourself.
Yes.
I see.
So on average,
I have a question, but maybe it's very basic. And,
who are you?
I'm Georgi Irdohin, student of Master of Bioinformatics. And thank you for the lecture. It was very interesting. And my question is maybe very basic. And nowadays, there are a lot of entries in UniProt, for example, which is annotated by homology and contains only AlphaFold predicted 3D model structure. And there are no information about binding sites and so on. And how do you think can I use your program Zetto and AlphaFold to maybe predict to retrieve information about binding sites for this type of proteins using, for example, interaction maps or so on?
There is a tool, I haven't used it, but I saw the paper, it's called AlphaFold. So there's a tool called that basically, I think, if I remember correctly, what they do is that I think they search PDB they take the structure from AlphaFold and they search PDB for similar structures and to look at the binding ligands for PDB and then to try to put them in there somehow. So there is a tool that should do something like that. And I think you can do it. You have to be a bit, the problem is in general, these long disordered regions. Because normally if you have a method that you look for cavities. And if you have a big loop that's standing up, it looks like a cavity. So you probably would have to cut away the disordered regions. And then after that, I mean, if you don't have any disordered regions, I think you could find it. So I'm sure you could predict like ion bindings and things like that quite easily, probably heme groups, things like that. Things on the surface are probably harder, but like, I haven't looked at it, but I guess if you look at the myoglobin model, it's going to have space for heme in the middle, I would assume. Because that's easy to learn, but it's so that, I think this AlphaFold does something like that. So I haven't, have a look at that and see what they do.
Okay.
I think there's a paper in bioRxiv, and I think the method is available, but I never used it.
Okay. Thank you.
So there was a question in the chat also here. So what was the minimum CPU and GPU requirements for running on AlphaFold? I think you can run it on basically a graphics card with, I think you would need 8 gigs of memory probably. And CPU, I don't think it's that hard. Probably anything, probably need like four cores or 8 cores of that, but it's really, and if you only have 8 gigs of memory, you're going to be some limited in size, but for normal protein, that should work fine. So I think basically, even a 1070 or it's, I don't know what the cheapest card with 8 gigs of memory. You probably can run 4 gigs of memory also in RAM, but that probably puts very small things. But the 8 gigs of memory card you can get for $500 or something like that. So
okay, thank you.
Oh, yeah.
Okay, if there are no other questions.
And I get lunch.
Yes. We can get lunch. I thank you very much for the.
So thank you very much.
Yeah, thank you.
Okay.
Thank you
And
for
I
talking.
remind everybody that the next lecture is on Monday morning at 11. Professor Tosato.
In the noise or
And
in the disorder.
And the disorder regions.
Okay. Thank you so much, Arne.
Bye-bye.
Be safe. Bye-bye.
Hope to see you in real life
Bye-bye.
sometime.
Many
Bye.
wishes. Bye. Ciao.
Ciao.